Hello.
Hello.
Hey. Where's Susan?
Hello, Susan.
Hello.
Hi.
I'm just, yeah.
Just
I'm just here. I haven't done anything this week.
all right.
Thank you for the paper, by the way, the mechanical.
Okay.
What paper was that?
What was it called? Mechanics of, no, I didn't. Epithelial cell stability based on mechanics? Something?
Yeah, something like that.
Yeah, I think I did have it, but it looks good, so
Okay.
I'll find it. It's in my other folder. It's in the downloads folder, of course.
Okay.
Well, Lalith, how are you? Hello, Morgan. All right. I don't know, Susan, did you have any updates on the things you're doing?
No, I've just been, like I said, cleaning. Very boring, but has to be done, so.
All right. And Lalith, do you have any updates or anything you'd like to talk about?
Hi everyone. So
Hi.
I kind of was working on the visualization, and I have, I've updated them this Friday to Bradley and a few other people, and yeah, I'm trying to understand how do I have to visualize the synthesions part. Now I have the labeled data that was given by the nickel Bhatla. So from that, can I use those labels? Can I do some analysis and do some kind of plotting based on my previous plotting? What I've done in the Friday week. So probably I'll try to mix those two things up and probably bring a nice update in the next week meeting so that I can show the difference. When we have the synthesium data and, suppose I have a synthesium and I'm trying to find it out and from the which close proximity and which lineage information it's actually aligning with. So yeah, I'll probably code it a bit in this week and I'll bring some good updates next week. So that's the plan.
Good.
As of now, trying out. And Gordon has been sending lots of papers and, it's kind of overwhelming at a point. Figuring out, studying and reading them, more like that.
Yeah, I try to keep into various aspects of the brain.
Yeah.
Okay. Susan shared a couple papers in the chat. The first is the mechanical stress driven by rigidity sensing governs epithelial stability. And then the second is molecular jackhammers eradicate cancer cells by vibronic driven action. Well, that's interesting. So that's an applied paper. Yeah.
I guess be careful when you're using ultrasound on cells. You might be breaking them apart.
Yeah.
Yeah, that may also apply to human embryos.
Mm-hmm. Yeah.
Okay. All right. Yeah, I want to get into a couple things today. The first is talking about something called Lagrangian structural tools that we can use to look at cellular systems and look at dynamical systems. So this is an example of a Lagrangian coherent structure, which I'm going to talk about. And this has been applied in fluid mechanics, and it's been applied in applied physics. But, it hasn't really, it's found different application domains. And until very recently, it's been semi-useful for biology. But people have been publishing interesting papers on how to apply this technique to developmental biology, and particularly embryogenesis. So this is an example of a Lagrangian coherent structure the idea is you have this turbulent flow, and in this turbulent flow, you have these structures that form. If you put in a tracking dye, or if you have a bunch of particles that are diffusing through a liquid, and it's this turbulent flow, you can get these structures that are,
What does coherent mean?
well, the coherence is that they form patterns, or they form these. I guess an incoherent structure would just be something that's kind of drifting along and not concentrated like some of these are. So you have these examples here where you can identify the structure.
Those change over time, I assume.
Yeah, they evolve and change, but the point isn't that like they exist and are pleasing to the eye. The point is that you can use mathematical tools to make a statement about the structure. So there's this,
Some of that flow will depend on viscosity.
right.
Yeah.
Well, they do this in different types of flows, and the idea is that mathematically you can identify a ridge, and those ridges kind of move over time and they diffuse, and you end up with different measures that you can use to characterize them, and they're related to the Lagrangian mathematical tools, as we'll see. So that's kind of what that looks like. And then this is the Wikipedia article on Lagrangian coherent structure. So this was something that was discovered by George Haller's group at I think MIT in the mid-aughts, and they kind of have been developing this cottage industry where they've been working on different application domains for this. And so Lagrangian coherent structures, or LCSs, are distinguished surfaces of trajectories in a dynamical system that exert a major influence on nearby trajectories over a time interval of interest. So usually you have like some origin point 0, and then you might have a diffusion of particles, and those particles diffuse outward. And you might have this turbulent flow that takes up the particles and they move around, and they end up forming these clusters or these ridges that diffuse outward. So if you think about like throwing like fish food into a pond, it usually just falls and then diffuses away. If this were a turbulent flow, you'd throw it in and it would follow the turbulence and would form these ridges where you'd have concentrations of the fish food. If you think about ocean gyres, it's kind of a similar thing, where the ocean gyres aren't turbulent, but they have this flow that, where if you put something in the ocean and, if something ends up in an ocean gyre, it ends up circling and circling the same part of the ocean, and it gets trapped there because the flow is sort of governing its movement. And then eventually you can get things like garbage patches where you get all sorts of plastics kind of concentrated in the same place. It's a function of the flow that you're in. So this is these are individual trajectories in a model flow. If you look on the right, and this is these are these kind of concentrations of things. We can use a model, which is using a Lagrangian to describe it, and you can also do this experimentally, where you use the Lagrangian, but you don't use it to sort of form you observe the trajectory instead of modeling it. So that's kind of, you start with this initial condition, and you end up going down these paths. And so there's a sensitivity to initial conditions. So you start off at a certain location, and you measure from that initial condition out to the current position. So it's like any dynamical system. You can have a discrete version and you can have a continuous version, and you measure out from the initial condition out as many time steps as you would like, and you can observe the evolution of the system.
Okay, sensitivity to initial conditions is a definition of a chaotic system.
Yeah. So it does. So if you're in a turbulent flow, you have the potential to have a chaotic system there. It sets up the conditions for that. So yeah, it very much does feed into that. Although I'm not sure how much people have kind of made the connection between LCSs and chaotic systems, or things on the edge of chaos or whatever.
Okay, taking that into account, it says attracting model LCS. Are those in any sense attractors like in chaotic systems?
I don't know what they mean here exactly, but I guess you would have sort of attractor points in the system. So like in a dynamical system, usually you have an initial condition and then there's some attractor points that can be described. Similarly, if you had a flow that was, I guess if you had a deterministic flow, you definitely have attractor basins or attractor points. If you had a turbulent flow that was maybe chaotic, you might still have attractor points. They actually talk about saddle points in here. So you're talking about, yeah, these stable points or these attractor points. Okay. So this is kind of talking about how these are used. And so basically what you do is you have this initial condition, you have these conditions of evolution, you're measuring the system using a Lagrangian, where you're describing the system using a Lagrangian and you're measuring the evolution through this trajectory. And you end up with this skeleton of the overall dynamics of the system. So you're basically, in the figure up here, individual tracer trajectories, which is where they might put a dye in a liquid or they might put a dye on some particles and look at the trajectories. These trajectories form coherent patterns that are generally sensitive with respect to changes in their initial conditions and the system parameters. In contrast, the LCSs creating these trajectory patterns turn out to be robust and provide a simplified skeleton of the overall dynamics of the system. The robustness of the skeleton makes LCSs ideal tools for model validation, model comparison, and benchmarking. LCSs can also be used for nowcasting and even short-term forecasting of pattern evolution in complex dynamical systems. So that's maybe a semi-connection to chaotic systems. Nowcasting, I'm not really sure what that is. It's a new term for like knowing what the current state of a system is or monitoring things in the environment. And then short-term forecasting is knowing how something is going to unfold in the short term, as you can't necessarily say things about the long term in these kinds of systems.
Sounds like Canadian weather predictions.
Yeah. It's basically I guess it's an improvement on some of the techniques that we've used to look at dynamical systems, and certainly is it gives you a very strong mathematical framework. So physical phenomena governed by LCSs include floating debris, oil spills, surface drifters, and chlorophyll patterns in the ocean, clouds of volcanic ash, and spores in the atmosphere. So a lot of things that you might expect for, measuring collective behavior or measuring things that are kind of in a fluid. And so this just goes through some of the mathematics of it and how this is characterized. So that's a little bit about that. This is a paper that is newer, and this is called Objective Eulerian Coherent Structures. So this is kind of another. So this George Haller was, sort of described these in the mid-aughts. So this is from 2022. Actually, I guess it's 2015, but also 2022. I don't know how that. Maybe it's an updated version. But anyways, this is on a different tool called Eulerian Coherent Structures, or Objective Eulerian Coherent Structures. And this is using an Eulerian. So the abstract here reads, we define Objective Eulerian Coherent Structures, or OECSs, in a two-dimensional, non-autonomous dynamical system, and instantaneously most influential material curves. OECSs are stationary curves of the averaged instantaneous material stretching rate or material sharing rate functions. So they're able to look at these in terms of materials instead of in flows. From these objective frame invariant variational principles, we obtain explicit differential equations for hyperbolic, elliptic, and parabolic OECSs. So they're linking this to different types of differential equations to describe these sorts of phenomena. This is not only in flows, this is in active materials. As illustration, we compute OECSs in an unsteady ocean velocity dataset in comparison to structures suggested by other common Eulerian diagnostic tools. We find OECSs to be the correct short-term cores and observe trajectory deformation patterns. So they kind of get into some of the tools that are used, talking about LCSs. So just talking about coherent structures more generally, which look at pattern formation in unsteady flows. They've looked at variational techniques in terms of how they're used to identify Lagrangian coherent structures. So this is prior work. And then this is talking a little bit about how this is being generalized, and then using an Eulerian as sort of a stand-in for that. So all LCSs, however, are intrinsically tied to a specific finite time interval over which they exert their influence on nearby trajectories. So in LCS methodology, you have an initial condition. The system evolves to these structures, these skeletons, and then they have this influence on nearby trajectories, and that only, it's kind of short-term prediction. This influence is an integrated one, filtering out short-term anomalies in the flow. Yet short-term variability in material structures is often seen as significant in highly unsteady flows, explaining the popularity of Eulerian, or instantaneous velocity-based diagnostic fields in fluid dynamics. So Eulerian methods build upon the LCS methods that they described earlier and that are being applied to systems and give this short-term description of the system. And Eulerian methods kind of build on that and maybe give you perhaps a better characterization. So this is kind of the mathematical background here. And so this paper is, so there was a paper in 2020 that described this using Lagrangian coherent structures in terms of talking about maybe things in development. Okay, so this is, I don't think I have that paper today, but this paper talks about coherent structures and active flows on dynamic surfaces. And this is Serra, who worked with George Haller on some of this stuff. And so he's been busy applying this to developmental biology and some of the data that would describe different developmental systems. And we'll talk about that in another paper. So the abstract of this reads, coherent structures, or flow features that organize material transport and deformation, are essential to analyzing complex flows in fluids, plasmas, and active matter. Yet, identifying such structures on dynamic surfaces remains an open challenge, limiting their application to many living and synthetic systems. So, we talked about how you can apply Lagrangian coherent structures to fluid flows, to particles and fluid flows, and then the trajectories they take forming these structures. And then we talked about Eulerian methods, which kind of build upon that. And then now, we still have to think about how do we apply this to living systems or synthetic systems that have biological components. Here, we introduce a geometric framework to extract Lagrangian and Eulerian coherent structures from velocity data on arbitrarily shaped time-evolving surfaces. So again, these are surfaces, and these are kind of using these mathematical tools to describe, the time evolution of these structures. And, the idea is that they're flowing on surfaces. And this will become useful because, of course, when we're talking about fluid flows and particles, it's easy. When we're talking, say, about a tissue or an embryo, where you have cells that are migrating past one another on a two-dimensional sheet or, in a three-dimensional volume, then that's a little bit different, and we need to describe it in a different way because there are a lot of mechanical influences that are not just fluid flows, there are other things as well. So that's what we want to do. We want to extract these Lagrangian and Eulerian coherent structures from velocity data. Our method operates directly on triangulated meshes, avoiding global parameterizations while preserving objectivity and robustness to noise. So they're using these triangulated meshes to describe the system instead of like the typical just kind of free flow. And so applying this framework to active pneumatic vesicles, collectively migrating epithelial spheroids, and beating zebrafish hearts. So they have these three different systems. Of course, we talked about active materials and pneumatic organization, where you have things that are oriented in one direction. So you might have a bunch of rod-shaped cells that are all oriented in one direction. And of course, their flow is going to look different than the flow of smectic oriented cells that are oriented in all different directions. And so that's going to affect the dynamics. And you can describe, of course, the flow of a pneumatic cell sheet or a smectic cell sheet using this technique. Then, of course, we have epithelial spheroids that are migrating collectively, so they're all moving in different, maybe they're all moving together, or they're moving in slightly different directions. They're being influenced by noise, so we want to be able to describe those dynamics. And then beating zebrafish hearts, where you have cells that are, I guess, migrating cells that are stationary, and you have movement in the system. So you want to be able to get an active description of those different types of systems. So using these different types of systems, they uncover hidden transport barriers and Lagrangian deformation patterns, such as dynamic attractors, repellers, isotropic and anisotropic strain. So they have, of course, the attractors, the repellers, so places where things are sort of stable or equilibrium points, points that are avoided by the system. And then you have these different ways to describe strain. And this is these are all things that are missed by conventional Eulerian analysis. So this is something that is added to that description. This approach offers a new perspective on soft and living matter, revealing how geometry and activity can be harnessed to program synthetic materials, and how Lagrangian strain and principal deformation directions can help uncover mechanosensitive processes and directional cues in morphogenesis. So I know Oregon is interested in the synthetic materials and programming synthetic materials. So this is kind of interesting that this is a tool for that sort of thing. Yeah, we're interested in these active matter flows, where we're interested also not just in active matter flows, but on curved surfaces. So when we're talking about the Lagrangian coherent structures, initially we were talking about maybe like a two-dimensional surface, although if you're talking about a fluid flow, it's usually a three-dimensional surface. But we want to be able to describe this on a curved surface. So if we have like a curved sheet of epithelial cells or something with curvature, we want to be able to describe that and not just the very flat two-dimensional case. And so this is useful in a lot of areas of biology and developmental biology. Recent advances in live imaging and computation have enabled the reconstruction of such flows across systems, ranging from developing embryos to engineered active pneumatics. So people are engineering materials that have these sort of pneumatic properties in them, and they want to figure out how, to get that, to get that sort of thing in engineered systems. These flows are spatially heterogeneous and temporally unsteady, driven by internal activity and influenced by surface curvature and topology. While kinematic velocity or trajectory data become increasingly available, its analysis remains challenging, yet essential for controlled material transport or discovering the underlying biophysical mechanisms. So they get into these coherent structures and their description. So they kind of classify Lagrangian and Eulerian coherent structures together, and these provide a reduced representation of complex trajectories, organizing material transport and deformation. And so they're actually, what they point out here, that coherent structures are typically objective, as we saw in the last paper. There's this objective Eulerian method. And so what they mean by objective is that these systems are invariant to the choice of coordinate system adopted to describe motion. So it's not dependent on the coordinate system, it's invariant to that. And it's robust to noise, which is useful because biological systems have a lot of noise in them. Okay. Coherent structures have been broadly applied in science and engineering. We've talked about this. In active pneumatics, recent work elucidated the connection between coherent structures and topological defect motion, revealing the interplay between flow structure and pneumatic order. So this is something where we have these descriptions of different types of skeletons in flows, and then we have topological defects and the role of motion. So a lot of times we want to describe topological properties of something, and we have mathematical tools for that. But we also want to describe maybe things like defects or things like movement and some of the noise around those sorts of things. So these are great tools for kind of modeling the idiosyncrasies of life and living systems.
They forgot something. These structures are often in tension. If they're a confluent tissue, if they're attached to each other, then they're in tension.
Okay.
So that's sometimes they're not attached to each other in an embryo. It varies.
Yeah.
But they're often a tension structure.
Yeah.
Which adds to the dynamics.
Right.
Yeah, Bradley also made a statement that there's a lot of noise in an embryo.
Yeah.
Has anyone actually studied that?
Oh, yeah. I mean, I don't have any papers on me right now, but there have been a lot. Now, I don't know if you mean in the individual cells or in the entire embryo. The entire embryo may be, well, I guess, yeah, there are papers on that, like with respect to.
Okay, are there papers on noise correction?
Yeah, I think so as well, but it's like I think there could be more work in that area. Let's just say, but yeah, I think there are. We'll have to put together some papers on that, and this is at the level of the embryo.
Yeah, I mean the closest we've got to that is variation between twins, for example.
Yeah.
And has that been quantified at all? And does it fit into these flow patterns at all?
The only thing I've seen things on noise correction. The one that's coming to mind is where they're doing stuff with positional information and looking at noise and positional information and things like that. But I'm sure there's probably stuff I don't know about in terms of twins or things like, or like get variation with the same genotype, I guess is what you're saying. Yeah, I don't know. I have to look in the literature for that.
No, I think one term that's a very old one is fluctuating asymmetry.
Yeah, that's okay. So, this is a paper that so this is Terajima et al. Multifunctional physical reservoir computing in soft tensegrity robots.
That actually applies to what we're talking about.
Okay.
It's a method of looking at tensegrities.
Oh, okay.
Which is the tensioned structure, which cells are if they're a confluent tissue.
Right.
So this applies. It's just that I've never heard of physical reservoir computing before. So Anybody know anything about that? What
Nope.
nerd?
Well, I think the reservoir computing is where you have like a black box, and you have a bunch of it's like a neural network. You have these neurons, or just like nodes in a network, and they're all connected randomly. And then you feed data into it, and it kind of sorts it. It self-organizes and sorts the data, and it produces this output. So it's like a technique people are using, sort of as a rival to neural networks. And it's hard to really describe what it's actually doing, but it produces some interesting results. And I imagine the physical reservoir networks might be like physics inspired instantiation of that. I'm not really sure what the physics part of it is, but that's what our reservoir networks are, and they're really, and I think hard to really grasp what they're actually doing. But so yeah, those are things to look into. So yeah, this is just kind of one technique, and they're kind of casting it onto the data in a sense. But it's interesting what they're kind of doing with it. It's really early days for this. So let's see, where are we here? So they also talk about active pneumatics work. Then they talk similarly Lagrangian coherent structures revealed new cumulative deformation patterns and dynamic boundaries of multicellular flows, robust to noise and experimental variability, while remaining undetected by Eulerian quantities. These structures provided new insights into morphogenetic mechanisms, pattern formation, and selfie bifurcations. While existing techniques cover general 2- and 3-dimensional flows defined by sparse, noisy trajectories, or flows on static surfaces, they remain unavailable for flows on dynamic surfaces, which are, of course, defined biology. So they actually have their code here in MATLAB and Python. And so these are some of the images of how this looks on a curved surface. So you have these Lagrangian coherent structures showing this geometric skeleton of these flow fields. So you have these spheres. You have this at time 0 on the left, time t divided by 2 on the right, on the second to the left, then time large t, and then the Lagrangian deformation. So you get this. These black points. These are the unsteady velocity fields. So you have them pointing in a certain direction. So you can see that the black. Let's see if I can zoom in. These black lines are velocity fields. So the arrows are going in a certain direction, and the direction is changing as you go along the sphere. Okay. And then you have these red. So you have the Lagrangian deformation grid, which is in red. So this is the grid here. It's this network, and it doesn't look, random, and it doesn't look regular. It follows these flow fields, so it's describing the flow fields, and it's showing that. All right, so that's in red. And then if we look at, so it's a growing spherical surface. The sphere is growing from T0, and so we have this deformation grid. We have these velocity fields. And then in B, which is Lagrangian deformation, which is on the right, we have an example of a Lagrangian coherent structure, or a set of these, marking regions of maximal material contraction or attraction over 0 to t, that interval. So this is where we have low levels, of material attraction, which is material contraction, and then high levels of material attraction. And you can see that, like in the low levels, the skeleton, or the Lagrangian deformation grid, is sparse, and then the areas where you have a lot of material attraction, you have a very dense deformation grid. So that's what this looks like. That's what they mean by deformation. It's kind of deforming the grid to conform with material flows on the sphere.
Okay, now Bradley, if they ran that twice, would it look the same?
Well, probably not, because the unsteady velocity field would change. so it might look different. I'm not really sure, but I know it's not deterministic. So
Okay, has anyone investigated under what conditions it would look the same?
you'd have to make the velocity fields deterministic. But in that case, I mean, if you're growing it at a steady rate and the velocity fields are deterministic, then it doesn't give you, I mean, it gives you information the first time, but I mean, I don't know if it would look any more regular. It might look regular, not sure.
Well, I'm just trying to imagine what it would take in terms of feedback or interactions between these points to make the result look similar.
Okay. Yeah, I don't know.
Okay. So, in other words, why did two embryos from the same brood look the same?
Right. And maybe it has something to do with the interactions between the velocity field.
Okay, so that's really not investigated at this point.
Yeah. So the deformation grid, I mean, I'm trying to think of the analogy of an embryo. It would be kind of like signaling molecules or something that would be sort of conditioned by a flow field, and it would kind of determine pattern formation or something. And if you had a deterministic flow field or deterministic concentration of different molecules, you would get very similar results.
Yeah, see, that's where things like bioelectricity might come in, because bioelectric fields are long range and therefore can organize a significant portion of an embryo.
Yeah, I think that's true. And of course, the bioelectric fields have their own dynamics, and they could be described with like an Alicea structure. I mean, the math would work basically. You would just, instead of having a fluid flow, have an electrodynamic flow. And you'd probably have to change the equations a little bit for it.
You could have both.
Well, yeah.
Okay.
So then they go through the math of Lagrangian coherent structures on dynamic surfaces. You have the Lagrangian here, which is described by this time evolution from t0 to t, and you get this integration, which gives you your f. And then there are these different measurements, FDLE and FTLE, which are the different measures of the coherent structure from the initial condition out to this sort of ridge that forms. And so they're different. You have like a continuous measure and a discrete measure, and they modify it for dynamic surfaces so that it describes sort of this curvature instead of describing straight lines. And so, they talk about they have to define the deformation experienced by a material patch in the neighborhood of x0, and this is this B term here. And then they talk about materials or maximal stretching, which corresponds to the largest singular vector of this B component. And you can quantify an isotropic Lagrangian deformation using this term here. So there are a lot of ways that we have to kind of change our description of these Lagrangian coherent structures, or these different measurements of that, to get to this curved surface, and in talking about deformation and things like that. This figure is Lagrangian deformation and coherent structure flows on dynamic surfaces. So this is interesting in the sense that we have some actual images we can look at here. Okay. So this is in A, we have a tissue flow. So we have this surface that's deforming, and we have a flow over that surface. We have a pneumatic flow. So this is an active flow on a dynamic surface. And this just shows how you have these active flows on dynamic surfaces. So flows are represented by three-dimensional velocities, V, on evolving two-dimensional surfaces, M. So we have these flows evolving on the surface. And then we have these two-dimensional surfaces that are deforming. We see that in B. we have this three-dimensional coordinate system which describes both the surface M and then the patches, which is described here on the top as XO, and then that evolves to XT. So you see that the description here, the F component from our Lagrangian description is described on this surface here, which is not deformed, where it's slightly deformed, and it's described as X0, and that evolves to this XT, which is this patch that's now deformed on the surface that's evolved over time. So this is basically this description, but on this sort of deforming surface. Okay, so then C is the material deformation experienced in the neighborhood of XO, which is this neighborhood here that we measured with our F component. And this is where it's encoded in this B component. So the purple patch here is encoded in this B component, which describes this area here. So this area is expanding within the patch. That's deformed. So this deforms material fibers. And then D is where we get these attractors and repellers. So we get this sort of anisotropy within the sphere. So we get these points where things are attracted to. We get these skeletal structures, and then repellers where we get things that are sparse, and there are very few descriptors in that area. So we have our repellers and attractors. We start to get that. Then we get the velocity field and trajectory things where we can describe the deformation and migration and these dynamics using a discrete mesh. Yeah. So that's basically that. And then they talk about Eulerian coherent structures. So they talk about that as sort of a longer term description of the same thing. Okay, so that's enough on that paper. One thing I want to talk about before I get to the last paper is this concept of action. And this is in physics where we have this scalar quantity called action. And so in physics, and I'm just going to use the Wikipedia article for, for the sake of simplicity here. In physics, action is a scalar quantity that describes how the balance of kinetic versus potential energy of a physical system changes with trajectory. Action is significant because it is an input to the principle of stationary action, an approach to classical mechanics that is simpler for multiple objects. Action in the variational principle can be used in different physical domains. And so in the simple case of a single particle moving with a constant velocity, therefore undergoing uniform linear motion, the action is the momentum of the particle times the distance it moves, added up along its path. Equivalently, action is the difference between the particle's kinetic energy and its potential energy, times the duration for which it has that amount of energy. So you can do that for sort of the kinetics, and then maybe what's more interesting for us is where we can look at the trajectory itself and look at the mathematical functional, which takes the trajectory, which is also this history of the system from the initial condition time 0 to whatever time we're looking at. We'll go out a number of time steps to get to this prediction, and we get this trajectory that describes the kinetics of the system. Which takes the trajectory of the system as its argument and has a real number as its result. Generally, action takes different values for different paths. So when you get different paths, and of course we know that we have this variational aspect, which means that we can have different types of paths. We have different types of paths for different cells, and sometimes we take the mean value of those paths. Sometimes there are different paths in maybe like an embryo. If we measure, say, 1,000 C. elegans embryos, if we look at the path of a cell as it migrates, and we find that overall it's basically the same, but there is variation in those paths. It's slight variation as it is, but it does kind of give you this variation to deal with. And so, we want to be able to understand that in terms of the action, And action ends dimensions of energy times time or momentum times length ends, and we do this for units of joules per second. So this is something that we want to think about, not just the kinematics, but the kinetics as well. So that brings us to the last paper, which is an application domain in developmental biology. Like I said, I don't have the 2020 paper on sort of the general application of this to developmental biology, because I thought that was kind of a broad overview, but this is actually a very focused overview of vertebrate gastrulation modes. So this paper is a mechanochemical model that recapitulates distinct vertebrate gastrulation modes. So this is applying this LCS methodology to a specific embryo and a specific thing in development. So the abstract reads, during vertebrate gastrulation, an embryo transforms from a layer of epithelial cells into a multi-layered gastrule. This process requires the coordinated movements of hundreds to tens of thousands of cells. So there are a lot of cells that are moving, and they're have to be coordinated, and we want to be able to describe that. In the chick embryo, patterns of actomyosin cables spanning several cells drive coordinated tissue flows. So we have these actomyosin cables that are intercellular or intracellular, and they're coordinating these tissue flows. And so we want to be able to describe those flows in terms of maybe like a skeleton or these sorts of structures. Here we derive a minimal theoretical framework that couples actomyosin activity to global tissue flows. We want to know these actomyosin cables, how are they contributing to the global flow of tissue. Our model predicts the onset and development of gastrulation flows in normal and experimentally perturbed chick embryos, mimicking different gastrulation modes as an active stress instability. And so this means that we have a change in how these actomyosin cables are behaving and how they're sort of, doing different things. Varying initial conditions and a parameter associated with active cell ingression, our model recapitulates distinct vertebrate gastrulation morphologies, consistent with recently published experiments in the chick embryo. Although our results show how changes in the patterning of critical cell behaviors associated with different force-generating mechanisms contribute to distinct vertebrate gastrulation modes by a self-organizing mechanochemical process. So this is a figure here where they talk about these actomyosin cables. This is figure 1. And this drives the tissue flow and gastrulation. So this is where we have these different structures forming the primitive streak. This is an A. So A is this PS formation diagram. And then in B, we describe the velocity. So what we're doing is we're describing these velocity fields. In the chick embryo, we want to describe sort of the speed at which the cells migrate, and we can measure velocity as a component of XY position and time. And the convergent extension of the mesoendoderm, red, which is red here, generates macroscopic tissue flows on the surface of the embryo. So as this red zone is changing, it's mesoendoderm, and it's kind of squeezing together and pushing outward like that. You see that in figure A. This is changing this flow field. So you can see the flow field is changing in terms of velocity. C at HH1, which is this first time step, the mesoendoderm sickle territory is characterized by supracellular cables. So this is the sickle formation here. And then you have the formation of these cables that they describe. These are supracellular cables with active myosin, phosphorylized myosin light chain, or PMLC. And this is perpendicular to anterior posterior, so it's going in this direction here, across the sickle from left to right. And then, let's see. Okay. Where are we? Okay. And then at HH3, 12 hours after HH1, which is this thing on the right here, where you've got this squeezed in the opposite orientation, the PS is characterized by supracellular cables with high active myosin, or PMLC, perpendicular to the midline. So now this is perpendicular into the midline like this, but this region has changed its sort of orientation. The pattern of actomyosin cables evolves from EJK XIV to HH3. Bar lengths represent the PMLC anisotropy, and bar. So they're referencing EKG XIV is down here in E, which is where you see these actomyosin cables, and they have the orientation and the intensity. And so that's these different parameters here. And so we're measuring this as kind of in the same way we're measuring velocity. We're measuring this XY coordinate system with time. So we're looking at the intensity here of these actomyosin cables. And we can see that we have these intensities change with the shape of these structures. And so then that kind of shows a little bit of that evolution here. You start to get the primitive streak forming. You can see some stains in these images here in C and D. And then this tells you the relationship between these equations. So the velocity is related to the orientation, and the orientation is related to the intensity, and the intensity is related to the velocity. So they all kind of play upon each other. So orientation, velocity, and intensity all play a role in each other's, in their dynamics. Okay, so then they talk about the mathematical framework. This is a predictive model linking mechanochemical activity and tissue flows. And so for a minimal model, we want to have a planar mechanochemical predictive model for formation or PS formation during gastrulation. So we have these three coarse grain fields, the tissue velocity field, which is V, and then these other components within it. The active stress intensity M, arising from myosin activity, and the average cable orientation, which is described here. So we talked about those in the last figure. In the limit of slow viscous flows associated with morphogenesis, we can neglect inertia so that the local force balance reads this, where the total stress is the sum of the viscous and active stresses. The viscous stress is where P is the pressure, I is the identity matrix, and U is the shear velocity, and this S component, which is a deviatoric rate of the strain tensor. The active stress associated with actomyosin cables is this term, with the components of B given by this term here. So they model the sheet-like embryo in two dimensions, as they described with their X and Y coordinates. This is a compressible fluid that allows for cell ingression into a third dimension. Okay, so that's a little bit about that. Then they kind of talk about they have this definition of stress here in this figure, which is where we have stress as force balance. So we have the viscous component where we have shear and dilatation. And then we have active, which is on this side of the plus sign, this active component, this viscous component on the other side of the plus sign. So that's the force balance. Then the other component of stress are the dynamics of active stress orientation, where we have this advection term here, we have rigid rotation, and we have shear-induced rotation, and that's additive across the system. And then we have the dynamics of active stress intensity, which is where we have advection, recruitment, dissociation, saturation, tension propagation, which leads to the induction of M, which is here. So that's all sort of this active stress intensity dynamics. Okay, so, and then of course they quantify the spatiotemporal morphogenetic flows, where they use their theoretical model. And this is where they get into this Lagrangian coherent structures methodology. And this paper I talked about from 2020 describes this kind of tool called dynamic morphoskeletons. So this is not something I'm covering today, but if you want to know, it's citation 44 in this paper. These are these dynamic morphoskeletons, which are basically these Lagrangian coherent structures that describe morphological structures in embryos and in other developmental systems. And so the DM is based on a Lagrangian description of tissue deformation captured by these finite time Lyapunov exponents, which are FTLEs. So I talked about how you have the finite, the FTLE and the AFDLE, which are two tools. So you have the finite time Lyapunov exponent, and I think it's the finite discrete Lyapunov exponents as well, or something like that. So there are these different measures of the Lagrangian coherent structures. Now, in terms of the FTLE, that type of measure combines local and global mechanisms along cell trajectories. So as we saw, we're measuring from the initial condition to some endpoint. That endpoint describes like a ridge, and those ridges describe the skeleton. So that's, local and global mechanisms. The DM consists of attractors and repellers towards which cells converge or diverge over a specific time interval t. So this is from initial condition to the time of interest. Repellers are marked by high values of the forward FTLE, while attractors are marked by high values of backward FTLE. So you have this forward and backward component, and this is kind of defined by these two parts, or these two regions of the phase space. And then attractors are marked, in their domain of attraction by high values of the backward FTLE, displayed on the initial cell populations here. So they give this, so they have supplemental figure 6, which I'm not going to get into, but that illustrates it. The DM reveals the organizers of spatiotemporal trajectories and is robust to noise, hence it is an ideal model for quantifying morphogenesis and comparing models with experiment. So this just kind of shows active forces in the chick embryo, where they have this experiment and model comparison. Then you have this developmental perturbations of gastrulation, where they perturb the system. They have, I guess this is, maybe this answers your question, Dick. So they have spontaneous twin perturbation, repeller and attractor for model velocities and experimental velocities. So this is like a twin model where you have repellers and attractors, and this is an experiment where you have repellers and attractors. This is VEGF inhibition, where they inhibit VEGF, which is a molecular signal for development for gastrulation, and you can see that it changes things in the morphology. You have FGF2 addition, so you add FGF2 in, and then you have BMP and GSK3 inhibition, which are other molecules that are involved in this process. And when you inhibit BMP and GSK3, you really lose any sort of morphogenesis. It just becomes this big blob. And you see this in the experiment, you see this in the model. So the model is more idealized in the experiment, but it's replicating what you see in the experiment. So this is just an example of how it can predict these kinds of patterns and dynamics. And so then they go towards a phase space of gastrulation flow patterns, where they want to provide a phase space description of gastrulation-related flows. So to go beyond studying the gastrulation modes, we use chemical inhibitors to uncouple cell intercalation and aggression and perturb the initial patterning and function of the prospective mesendoderm, as described in a recently published experimental paper. And so that shows that we can recapitulate these phylogenetic variations in the morphodynamics of vertebrate gastrulation, where you're coupling the dynamics of the actomyosin cables to large-scale tissue flow patterns. So they've done this in experiment, and they wanted to sort of replicate this computationally, or replicate this using this kind of phase-space dynamical systems. Okay, so they basically can make successful predictions, and this is a figure here of the evolutionary transitions and gastrulation patterns. So they go big for the final figure, and they show that, like, if you want to, they had a heading in there that said reptilian mode. What they're doing is they're doing the experiments in chick, doing the modeling in chick, and then they want to look and see if they can do this in reptiles as well. So in the chick, you have a primitive streak because it's in birds, and then in reptiles, you have the blastoporal canal. So that's what they're looking at. They're looking at these two homologous structures, and they're looking at their formation and experiment and model. So they're actually looking at the initial condition. So you have the sickle modeled here, and you have this sickle where there's a reduced mesendoderm, and then you have this at T0. This is the initial condition. This is the initial condition for amphibians and teleost fish. So in amphibians, of course, we have this blastopore. In teleost fish, we have the germ ring. So again, another part of this vertebrate clade where we have chicken reptile here, and then amphibian and teleost fish here is sort of the outgroup, or the outgroups. So we have this sort of ancestral condition here where it's this ring. We saw that ring before, but it was like where we inhibited certain factors in the chick embryo. Now it looks very similar to what we see in what's shared between amphibians and teleost fish. And so we use that for our ancestral condition there, or initial condition. And then we start to look at the evolution. So we look at how this process unfolds in these four taxa, this developmental process of cell ingression and cell intercalation. So you can see there are different patterns of it throughout. And you can describe this with this mathematical model, but you're also, what you're doing is you're changing the conditions under which this migration happens. Okay. So I think that's all for that. Any questions, any comments, any inspirations? What do we have to say?
I guess I just wanted the reference for the first paper you went over, because the different flows.
Yeah. This was this one here. Coherent structures and active flows.
Yeah, I think so. It was the first one. I missed the reference for it.
Right, let me see if I could get, this is a preprint bioRxiv. We get the chat right there. So that's the bioRxiv link, and then this is the title here. I'm not sure if this is out in a journal. It's pretty new, so probably not yet. But it's,
Okay. I'll copy it over and look for it.
okay, yeah. Any other thoughts?
I think flows have a lot to do with embryology.
Oh, yeah. Okay. That's all for today. Why don't we call it a day? See you next week. Thank you.